Hello everyone, my name is Chris, and today I will be going through how I attempted some DIY ecology research on some local barnacles and mussels. Now, before I get into things too much, I should probably give a brief overview as to exactly what ecology is. So, ecology is sort of the study of how different animals or species or other living things are distributed, and essentially where they live, how many of them are there, and how these different species interact with each other to form different communities within their environments. And so we often want to study the different living, so biotic, or non-living abiotic factors that affect how these different species are distributed. So some major different types of interactions that ecologists like to study are things like competition, where different species are competing over a limited resource, something like space. Or there can be things like predation, where you have different species that are actively consuming each other for sustenance. Now, a classic example of how these different abiotic and biotic factors affect how species are distributed, that has been talked about in both of my ecology classes up to date, is the example of barnacles living on rocks. So now some basic background information is that along beaches, the tide rises and falls throughout the day. And as such, different parts of the beaches are covered with different amounts of water for different periods of the day. So many species prefer to be lower in this intertidal zone because that way they are covered with water for more of the day, and the water is sort of what provides them with all the food that they need. Whereas species that live further up in the intertidal zone, they're covered with water for less of the day, and as such have to deal with issues such as like drying out from the sun. As such, in these intertidal regions, there's often a lot of competition between many different species for access to space lower down in the intertidal zone so that they can get all the nutrients they need from the ocean. In 1961, Joseph Connell studied two different kinds of barnacles and their interactions living on rocks in the intertidal zone. He studied Semibalanus and Thamylus. He found that the largest factor impacting Semibalanus's range was the fact that when it got too high up in the intertidal zone, it would just dry up and die. Now, what limited Thamylus's range was in fact Semibalanus, because Semibalanus is the much stronger competitor. So in regions with the two barnacles present, fighting over space, Semibalanus will almost always win and knock out Thalamus. However, Thalamus is more tolerant to things like being dried out, and as such, it is actually found higher up on the intertidal zone than Semibalanus, because it can survive more of those harsh abiotic conditions. So here in British Columbia, we have two very similar species of barnacles. We have our very own Thalamus species, so this smaller, less competitively inclined barnacle, and we have our own larger Balanus barnacle. However, we also have species like Mytilus, or the blue mussel, and along with many other different marine invertebrates on these rocks. Now, between these species that I've talked about, Mytilus, or the mussels, are the most competitive. If the conditions are favorable for them, they will absolutely take over any rock. Next, it's Balanus, and finally, our weakest competitor today is Thamylus. So my research question was, how does the abundance and distribution of these invertebrate species vary across two different beaches in Vancouver? Now, how does one go about counting barnacles on rocks? Now, the most obvious and accurate answer may be to go, okay, I'm just going to look at every single rock on the beach and count every single barnacle there until I have the most accurate image of that beach. Now, there are a few issues with that. First of all, beaches are there's lots of rocks on a beach. It would be incredibly taxing and extremely difficult to be able to count every single rock on that beach and look at every single barnacle there. So, in situations like these, ecologists, we like to do something called area-based counting, specifically with quadrats. So what we'll do is instead we'll look at many different smaller chunks of the larger area and use that to approximate what we think the entire larger area looks like. Now, because by looking at many different smaller quadrats, instead of looking at the entire beach, there will be some differences, and we don't know exactly what these differences will be, but we have a word for it, and that's called sampling error. Now, there's different ways that we can try to minimize sampling error. If we sample more points, or we have more samples, then we'll get a better depiction of the beach. Because if I just took one rock and was like, okay, I'm just going to say that rock represents the entire beach, well, that might not be the entire case. But if I sample, say, 50 rocks, then I'm getting a much better image of what that beach actually looks like. So we also want the quadrats that we're measuring to represent the area around them as well. It would be really bad if I were just to go to the beach and then look at all the rocks with only seaweed on them, if there were only a few rocks that didn't have barnacles on them, because that way I'd be getting a really inaccurate depiction of what this beach would look like. So, one Friday morning I went down to Wreck Beach at 7 a.m. to count some barnacles, but things took a turn for the worse when I realized A, a quadrat made out of 2 shoelaces was a terrible idea, B, counting barnacles by hand was absolutely awful, and C, that my phone took absolutely terrible backup photos in the low light. So I went back to the drawing board. After reading a paper by Riccardo Scorsati and Martin Freeman and learning they used a 20 centimeter squared quadrat, I decided to tape 4 pens together and call it a quadrat. Next, I decided to also bring a little snail tape measure and a 30 centimeter ruler to take some additional measurements, which you will see why I used them later. Finally, I decided to use the DSLR that I had been used to film myself count the barnacles to instead take photos of the barnacles to count later so that I wouldn't have to count them by hand. So the next morning I went back to Wreck Beach and was much more successful with my data collection. And the following day I went to Spanish Banks and I got absolutely drenched, but I took those photos. While at Wreck Beach, I also got to witness some other forms of community ecology, most notably this prime example of predation between this here seagull and this unfortunate crab. And in fact, we even see a bit of intra-specific interactions going on between these 2 seagulls over the crab. Now, I guess an interesting thing that I visually noticed while visiting both beaches is that they were quite different in both of their geographies, because at Wreck Beach, all the rocks were actually below the high tide line, meaning that they would all be covered in water at some point in the day. But at Spanish Banks, the rocks extended above the high tide line, meaning that many of those rocks would just not be covered in water on a regular day. And as such, that meant that all the rocks at Wreck Beach had barnacles on them, but at Spanish Banks, there was sort of an abrupt cutoff line where some of the rocks had barnacles on them, and then everything past that there was no barnacles on them. And also while examining these beaches, I saw there was actually a whole ton of diversity there that you don't initially see when you're standing far away. And up close you can see so many different species like these little limpets, these small little snails, some oysters, you get plenty of seaweed, and you can see that it's quite a complex community there on these rocks, which from a distance seems so simple. It's quite amazing. Then, after all the photos were taken, it was time for the most tedious part, counting the barnacles. Now, luckily this was facilitated in part thanks to the technologies of Adobe Photoshop, so all I needed to do was just simply place a dot on every single invertebrate I saw, and then I could have Photoshop count out how many dots I saw and then report to me the totals. And then I could just plug that all into Excel. Now, I quickly ran into the issue of species identification. So in lectures, we had been told that the main difference between these 2 barnacles was that Thalamus was tiny and Balanus was big. But in the wild, you see quite a range of barnacle sizes, and that sort of threw me on a loop while I was looking at the photos. And then after trying to consult some online guides, I was just left even more confused. A lot of them just stated that it was really difficult to tell differences between barnacle species. So I did the only reasonable thing, and I went to my ecology professor, who is a certified barnacle expert. And he gave me a great guide on how to distinguish the 2 species. So essentially, Thalamus is the smaller barnacle, and it will never get above half a centimeter in diameter, whereas Balanus can grow much larger than that, but it can also be smaller than that as well. So size is not the best way to decide. However, a better way to determine the species identification is through looking at their shell and at their cover plates. So Thalamus shells are made out of 6 plates of equal size, whereas Balanus shells are actually made out of 4 large plates, and then these 2 very thin plates, which look kind of like ridges on the side there. In addition, the cover plates of Balanus are also quite diamond-shaped with sort of a jigsaw fit to each other, whereas Thalamus's, they're just quite rounded and lack that jigsaw fit. Another thing that I'd come up is I'd wondered if I should count the areas that the barnacles take up with each quadrat, and he gave me a great idea for that, which was to actually just assign random points inside the quadrat, and then see how many of those points touched a barnacle species, and then out of that total I could calculate a rough estimate of the percentage that each invertebrate took up within the quadrat. And that way it's actually a form of random sampling, which is super cool. So I did that, and I added it to the measurements. Now I don't actually go and do any of the statistics on the areas, but if I wanted to, I could do that too. And if you're interested in seeing all the data I collected, then in the link below you can see the spreadsheet of everything I did. So because I didn't have any direct way of measuring the heights that these barnacles were above the lowest point in the intertidal zone that I measured, I had to create a model. And this model I'm dubbing the CCBM, Chris's Crappy Barnacle Model, and it uses enough approximations to make a physicist cringe. Okay, so this is where we sort of get into the iffy side of things. So when collecting the data, one thing that I wanted to learn how these barnacles varied with their heights throughout the intertidal zone. Now, to do this, I essentially just approximated the beach as a triangle of sorts. Now, as you can imagine, beaches aren't, they're sloped, but I don't know if one triangle is sort of the best approximation, but it's what I'm working with here. So to make this possible, what I did is I tried to measure rocks at roughly the same sort of lateral points on the beaches. So I measured rocks sort of in a row on the lowest point of the beach, I measured rocks in a row on the highest points of the beach, and then I measured rocks in a row at a midpoint on the beach between the high and the low. And this, what it allowed me to do is it allowed me to sort of try to control for the height of the beach. There obviously is some minor differences, because what I could do with my limited resources is I could measure the height of the quadrat to the sand below it. And this very cool program from Fisheries and Oceans Canada, which is actually currently in its testing phase, like it's not properly released, but it's an interactive tide tracker, and it actually has predicted tide levels and the recorded ones, and for each timestamp. Look at that, 2 decimal places. Ooh, that's to the nearest centimeter. That's good. So what I did is I tracked the time when I took the initial measurements, which was at low tide, to get the height then, and then I came back a few days later and to when the water level was about where I took the highest measurements. And so by looking at these tide charts for those days, I could then determine, okay, how high was the water when I went there once and how high was it then there, and I could take the difference, and that gave me the height difference between my measurements, which is what I'm using as the beach height. So what I could do is I could take that difference, divide it in 2, and add that on to all the midpoint measurements. And then I could take that difference and just straight up add it on to all the high point measurements. And so combining all that, this is how I sort of got the height distribution from the lowest point of the intertidal zone to the highest point of the intertidal zone for the barnacles. So yes, this is CCBM, Chris's Crappy Barnacle Model. So this brings us to the fun part, statistics. So now that I've gotten all this data of all the barnacles and their various abundances within the quadrats, well, how do I know what this all means? And for that, I need to analyze it using statistics. So I've been taking a statistics course this term, and I thought it would be a lot of fun to try to use the knowledge that I've gained in that course and to actually apply it on some experimental data that I've collected. So for this course, we've been using a program called R, which is a programming language designed specifically for statistical use. Now, what's also nice about R is that it can also graph things for me. So right here, I can sort of see the scatter plots between the heights above low tide that I measured, and I can also see the amount of barnacles that were present in each quadrat, and those are for the 2 beaches there. Now, before I get into that, I should probably explain a little bit about how statistics likes to operate and some of its quirks. For statistics, we always like to compare stuff against something called a null hypothesis. So this is, in effect, the most boring explanation for a thing possible. So in this case, a null hypothesis would be that balanced counts and the height above low tide is not correlated. And what this allows us to do by having a null hypothesis is that we can be really specific about what we would expect to see if there was no correlation, because we'd expect the correlation to be 0, and therefore that is our null hypothesis. So with this information, programs like R and whatnot, they can construct something called a null distribution. And that is sort of what we'd expect the data to look like if this null hypothesis were true, that height and balanced counts were uncorrelated. And then what it's going to do is these tests, essentially, they take the data that I collected and it compares it to that distribution of the null hypothesis, and it sees, okay, well, what is the probability that the data that I collected falls into this distribution. So in a lot of sciences, we create sort of an arbitrary value called alpha, and what we hope for, and it's sort of an arbitrary value that was decided, don't know who or don't know why exactly, but it's 0.05, so 5% of the time. That means that if this null hypothesis were true, that our data would fall under it 5% of the time, which is, for all you D&D nerds out there, the same probability of rolling a nat 20 on that old D20. So generally, if we obtain a p-value, or the probability lower than 0.05, what we're going to do is we're going to say, okay, this is seems to be very unlikely that our data would fall under this null distribution, and so we are going to reject the null hypothesis and say that it is wrong. So when we reject a null hypothesis, it means there is some better explanation out there for why we see these trends. And we call these potential explanation alternative hypotheses. And we never confirm any of them. We just sort of go like, oh, I guess there is some better explanation, and it's probably hiding in one of these alternate hypotheses, but we need further testing to find that out. And alternatively, if we do not have the statistical power to reject a null hypothesis, such as in the data that I collected for Spanish banks between height and balance counts, as you can see, I got a p-value of 0.077, which is 7.7%, which is not low enough to reject the null hypothesis. So now, instead of saying we accept the null hypothesis, it is true, there is no correlation, instead, what we say is we don't have enough data to reject the null. So we never accept a null hypothesis. We only ever disprove it or say that we don't have enough data to do so. So those are just some of the quirks that statistics is based around. So for the data that I collected, I wanted to see a few things. I wanted to see if the heights above low tide had any effect on the counts of the invertebrates that I was looking at, and if there were any differences between the beaches. So I needed a test to compare the distributions between height and invertebrate counts and between the two beaches. So this wasn't something that we had actually covered in my stats class yet. So I went to my prof's office hours and I asked him, and he pointed me towards something called an ANCOVA test, or an analysis of covariance. And essentially what it is it's comparing these two scatter plots against each other to determine actually multiple things. It can help me determine does height play a role, and do the beaches play a role in determining the invertebrate counts. So I have one of these up here. So for looking at the Balanus totals, so this would be the height playing a role in the Balanus counts. You can see I got a p-value of 0.0065. So that's like 0.7% of the times that this data would fall under the null hypothesis. And so we can reject that. And basically what we're saying here is that in both these cases, for both Wreck Beach and Spanish banks, that it's clear that height seems to play a role in determining the Balanus counts, and that the relationship, the null hypothesis, that height and Balanus counts aren't related is garbage, and we can get that out of here. If we look here, if we're comparing the distribution between the two beaches, we can see we got a p-value of 0.0514, so that's just above 5%, so it's not low enough to be statistically significant. And so in this case, we can't reject the null hypothesis that the beaches have the same distributions. So what we're going to say here is we don't have enough data to say that these beaches have two distributions. And just by looking at the scatter plots compared against each other, we can see that the beaches generally do have fairly similar things. Like if you were to squint your eyes, and we can see that the similar trend lines and whatnot. So now I can go look at something like the thalamus barnacles. Once again, very similar to the Balanus, I got a p-value of 0.002 for the heights. So it's clear that height plays a role in determining the abundance of thalamus barnacles. But once again, looking at the beaches, ooh, that's a p-value of 0.21. So that means that this data would fall under the null hypothesis 21% of the time. And so we definitely do not reject the null here. So it seems like the beaches generally have similar distributions of thamellus barnacles. Now, Mytilus, this one, the blue mussel, this one was super interesting because just while I was even at the beaches, a stark visual difference between how many mussels were there. For at Wreck Beach, there were hardly any mussels, and all the mussels that I saw, there were often like little cracks between rocks, very few, whereas at Spanish Banks, the mussels were just everywhere. So this is going to be kind of interesting. So we can see here that the height above low tide does not seem to impact mussels abundance that much. It got a p-value of 0.23. So that's very high. So we definitely do not reject that null hypothesis. But we can see that between the two beaches, just looking at the abundance of mussels, I have a p-value of 8.056 to the minus 6. So that's a super low number. So in this case, we definitely reject the null hypothesis that these two beaches have similar distributions of mussels. To answer the big question on how these beaches vary in their distributions of barnacles and marine invertebrates, I can say that with my data that barnacles don't seem to differ much between Wreck Beach and Spanish Banks, but mussels seem to differ quite a lot. And there we are. But further testing is needed to determine the causal factors behind these differences in distributions. There's multiple different factors that could be affecting these distributions. For example, Wreck Beach. It's facing the west side, and it has like open ocean right in front of it, so maybe different abiotic factors, like maybe there's just harsher waves and the mussels don't like that. Or maybe because it's facing the west side, it gets more direct sunlight and the mussels don't like that. Whereas Spanish Banks, it's facing north. We have North Vancouver like just across the water from there, so maybe the waves are less harsh. Maybe it's just getting less direct sunlight, those mussels there, and they like that, and so they do better there. And I guess on this note, during the office hours talk with my ecology professor, I was talking to him, and he was telling me about how mussels, their growth is super impacted by sunlight, and that they are super sensitive to direct sunlight. And generally, I guess this is true for barnacles as well, but especially for mussels, they just do not like direct sunlight. So, I mean, this is not something that I measured during my data collection, but like if I were to go out again, I could like measure the angles of the rocks that I was measuring these mussels at and like what direction they were facing in. Now, for most of my data, I was collecting these quadrats towards the water. So at Wreck Beach, almost all my quadrats were facing west, and at Spanish Banks, all of them are facing north, but I don't really quite have the data to mess around with that and test that. But these are just all fun things to keep in mind for future experiments. So this is now a learning opportunity for the viewer. Yes, I am now encouraging you to get out there, count some barnacles, take out your compass, find them angles, do some trigonometry, and report back to me when you find the answer. Okay, yeah, this was a very fun project. Definitely a fun look into the a very basic and fun look into the world of ecological research. But yeah, it's very interesting, and yeah, I don't think I'll ever look at barnacles the same way ever again. But thank you all so much for watching. This was a very fun experience, and really like being able to apply a lot of the knowledge and stuff that I've learned in my classes, and to see some of these patterns in effect for myself that have been described to me. So yeah, tune in next week when I will be doing mark recapture to determine the number of silverfish present in my bathroom.